So welcome everyone to the last part of chapter 27 and even last part of all of actually these videos that I'm talking about, the chapters in Bio 201. I will try not to get too sentimental here, but it's been a pleasure. That's all I can say. It's been a real pleasure to do these videos and to teach all of you guys. So today I'm going to be talking a lot about prokaryotic genetics. So as we one thing we know is that prokaryotes do not reproduce sexually. They do not have sexual organs, right? So they cannot have sex. They cannot, create new babies through reproduction. But one thing they can do is they can actually transfer genetics among two bacterial cells or two prokaryotes. And this actually fuels, of course, evolution in quite some way because, of course, evolution is passing genetic traits, right? Well, here, prokaryotes can actually evolve because they can pass their genetic traits. If they were not able to do so, this would really stop a lot their evolution, believe it or not. So, three ways is how actually prokaryotic cells can actually, transfer genes through this horizontal gene transfer is conjugation, transduction, and transformation. So conjugation is actually more cell-to-cell contact, is when two prokaryotes create actually it's called actually a sex bridge. In some way, there's a small bridge and where genetic information is being passed. Think about this as very similar to what you see in plasmodesmata in two plant cells, is this connection that, two plant cells are actually connected with each other. Well, the same thing goes here in a conjugation, is that when these two prokaryotic cells are able to create this nice bridge that is able to pass genetic information. Then you have transduction, and transduction is basically mediated via viruses, which I'll be talking a bit later. And the last one is transformation. This is coming from the environment. All of these three processes, so these ones over here, the conjugation, transduction, transformation, they're seen in bacteria, and they also can be seen also in archaea. So let's start with transformation. It's a very, very easy one. So there's something called natural transformation. It occurs in many bacterial species. The biggest, most important one that this happens is Streptococcus pneumoniae. Remember, this was the one that causes this massive and annoying diseases in the lung, this pneumonia, and it's actually where Griffith studied them a lot. So one thing they saw is that Streptococcus is that when they die, they can release DNA. They will release it to the environment. And what happens is that another live streptococcus can take up that DNA. So it's quite interesting how this works, really, in a way that basically a dying cell throws its DNA and another cell can come and pick it up, just like that. So let me show you an example. Let's imagine you have here a cell that's dying. And basically, what's going to happen is going to push out its DNA to the environment. Then what happens is another live, basically, prokaryote, or in this case, let's imagine an E. coli cell. So imagine this is an E. coli cell. It dies. It throws or like literally pukes out the DNA. Another live E. coli will basically take it up, and what it can do is it can take this actual genome and integrate it in its own genome. So, now it's gained an extra DNA. So imagine that this E. coli has extra genes, which this one doesn't have. So it will pass it through transformation. So this is actually a very, very, this is the natural transformation. We see also what we've done is basically we can do something is artificial transformation. Is that some species of bacteria do not undergo transformation. They do not. But one thing we can do is that we can actually make them in the lab, push them to do transformation. And one example is E. coli. E. coli cannot do natural transformation. So sorry for using the wrong bacteria here. It's mostly Streptococcus that does it here because it can do this natural transformation, but E. coli cannot. But what I can do in the lab is that I can force E. coli to actually do transformation. So what I can do with E. coli is that I can actually break its membrane a tiny bit to create an opening and where I can then put DNA, any type of DNA that I want to basically for it to absorb. For example, let's imagine I have a piece of DNA that gives you the green color. It's called, let's say, a green fluorescent protein. So it's a protein that's fluorescently very green. So what I can do is I can actually take an E. coli, I can open its membrane a bit, using certain chemicals. And I can then basically add this DNA that encodes for this green fluorescence protein, and it's basically going to go inside the E. coli. So now the E. coli will actually be green fluorescent. So this is a very actually a good technique in the lab that you use a lot, and most probably you guys will be doing this in your labs. It's how sometimes we can actually do cloning of certain DNAs. We can push it into an E. coli, and inside the E. coli it can start to replicate. Now let's go to transduction. Transduction is done mostly by viruses, and specifically in bacteria, it's done by bacteriophages, because those are the bacteria that infect, sorry, those are the viruses that infect bacteria. So you have two types. You have generalized transduction and you have specialized transduction. Generalized transduction means that any type of bacterial genome can be basically passed on. Specialized is only certain types. So let's just focus right now on generalized transduction. Generalized transduction is basically any gene can be transfected or transferred, and it occurs through accidents in the lytic cycle. So as we remember, bacteriophages have this lytic cycle in where they will actually bind to a bacterial cell, they will infect it, and they will replicate, and then they will lyse it. Hence, lytic cycle. So it can happen that sometimes is that when the bacteriophage goes in, it can actually completely destroy the bacterial genome and by accident take a bit of it. I'll show you examples here more with certain pictures. Specialized transduction happens in accidents via lysogenic cycle. So we know that some bacteriophages, they do not go straight into the lytic cycle. They do not go and start replicating and destroying the cell. No, they will actually take their genome and actually kind of transfer it or kind of combine it together with the bacterial genome to create a prophage. So prophage is bacterial DNA plus the bacteriophage DNA together. So then what happens is then later is when, of course, now the lysogenic cycle is done and it goes to the lytic cycle, the piece of basically bacteriophage DNA is cut in a weird way that it actually takes a bit of bacterial DNA. I'll basically here again use very nice pictures to describe each one. So let's start here with generalized transduction. You have here, for example, this uh, bacterial cell, and it has basically this A gene, A plus gene, and B plus gene. Okay, let's imagine now a phage comes and infects. It basically, will attach. It will release its genome, and then here basically it's going to start replicating. And when it does, sometimes it will actually break the bacterial genome into pieces. So you can see here now basically everything is in pieces. This whole circular DNA that is found in the bacteria, it's in pieces. And then when basically everything is now replicated and now you produce new bacteriophages, it's going to, of course, start to take a bit of the bacteria, sorry, a bit of the genomes. So mostly they will pick up their own genome. So as you can see, the blue string is phage DNA, while the one basically shown here in red is basically more the donor, the more the bacteria DNA. So what happens is that, of course, these now newly created phages will pick up their DNA, but sometimes by accident they will take up the bacterial DNA because now it's in pieces, and by accident it takes it up. So, for example, in this case, it took the A plus gene. So what happens is that now this guy over here goes and infects another bacteria. It does the same thing. It injects, and now it's injecting just bacterial DNA. So this bacterial DNA, basically what can happen, for example, in this car now, this cell has negative B, negative A gene, negative B gene. So what can happen, it actually can change it through something called homologous recombination, and where this A is basically substituting this portion. So now this new cell has a new gene. Before it had only A minus, B minus, now it has A plus and B minus. And of course, this A plus came from another cell. So this is how this transfer can happen. This is generalized transduction, and it mostly happens through mistakes of the lytic cycle. Now let's talk about specialized transduction. This happens in mistakes of the lysogenic cycle. So we have here basically, again, a bacteria, and then you have a phage. The phage basically puts its DNA, which is shown in red, and you have the bacterial DNA, which is shown in this basically blue, purple color. So of course, as we know in the lysogenic phase, is that the phage DNA incorporates into the bacterial DNA to create the prophage. So this whole thing is now called a lysogen. So a bacteria is now a lysogen because it contains a prophage. So now when basically it goes back into the lytic cycle, what's going to happen is that the bacterial DNA, the phage DNA is going to basically remove itself and recircularize. But sometimes what happens, it will pick up a tiny piece of bacterial DNA by accident. So you can see here, this is red with a bit of bacterial DNA. So the red is the phage DNA, and this basically kind of purple, blue is this bacterial DNA. So it picks it up by accident. Now it's to replicate, and now you create phages that have basically both the basically the phage DNA and also the bacterial DNA. Where in this case it was purely red, it was just what's it called? Just just phage DNA. Well, here now it's both. So now imagine this phage is going and infect another cell. This guy over here is going to integrate and actually is going to transfer basically new bacterial DNA. So this DNA from this guy has now been transferred to here through an accident of the lysogenic cycle. That's called specialized transduction, and only certain genes can be taken. Here, as you can see, any random gene can be taken. Here, no, it's only genes that are close by to basically the phage DNA. So hence why it's specialized. And now comes conjugation, the last type. So conjugation is basically when you have certain, bridges that are being created between two bacteria cells and they're passing on plasmids. So remember, plasmids are these kind of small circular DNA. So you have this big genomes and you have small plasmids that are small pieces of DNA that actually encode for some sort of advantage. And I'll give you here an example of E. coli. E. coli sometimes have this F plasmid. So this F plasmid is called the fertility factor. It's a nice plasmid that gives E. coli pili. It gives these small little hair-like structures that helps them attach to places, but to also do conjugation, to do these bridges. So certain, let's say E. coli will have the plasmid, and we call them F plus. So they contain the plasmid. And other E. coli that do not have it are called F negative cells. They do not have these F plasmid, this fertility factor. So let's imagine how this conjugation occurs. Okay. So imagine here you have basically this is the bacterial genome, and this is this F plasmid, this extra circular DNA. It's very small compared to the genome actually. So this is the F plus because it has the F plasmid, and this is the F negative. It doesn't have the plasmid. Sorry for this, guys. Let's quickly remove this. Oh, perfect. Cool. Sorry. So you have the bacterial chromosome, basically the F plasmid. So now this guy over here, because it has the F plasmid, it can create this pili, these small little hair-like structures, and where it's literally going to go attach to this F negative cell. It's going to attach and it's going to create a bridge, as you can see here. And what happens is that now this F plasmid is rolling. It does something called rolling circle replication, in where it's gonna literally this piece of strand is gonna roll here, and while it's rolling, it's basically creating a new piece of DNA shown here, basically in purple. So it's rolling, so it's removing and passing it here, and basically it's kind of replicating itself, hence the rolling. So now that this strand is coming here, and now this other F negative cell, the recipient cell, it's basically replicating this new incoming DNA. So now both of these become F plus. So they have the basically the F what do you call it plasmid, and this F plasmid is what gives this pili. Because for example, this F negative cannot doesn't have the F pili because it doesn't have DNA encoding for it. Well, this one it does. It has the F plasmid. The F plasmid encodes for these nice pili that you can create these genetic recombinations. But sometimes, so this is just another example of what I just said. So I'll repeat this again. Why not? So you have, of course, this is the donor. So this is the F plus cell, which is the one that's giving. So the donor. So this is the bacterial chromosome. Nothing happens there. So then you have this basically F cell. So you open this basically mating bridge, the sex bridge, which is done basically mostly through basically this pili. And this pili basically now is going to help the basically transfer of the plasmids. So what happens here is basically now this F cell is going to pass its genome through this rolling circular replication. It's going to pass it. And while it's passing, basically this replication happening in circular motion, This passes here to the F negative cell and basically starts to create. So what happens sometimes here is that now let's imagine we come here to this basically cell. Okay, this plasmid can integrate with the chromosome. So now this F positive cell, which is basically like this here, has the F plasmid. This can now integrate inside here. So they have this insertion sequences, so the sequence that helps them basically integrate. And now when it integrates, you create something called HFR cell. HFR cell is called high frequency of recombination because now this can actually recombine a lot with other genes of other basically cells. So of course this is called integration, but what can also happen is now this integrated F plasmid can also then excise and remove and basically become a plasmid again. So this gives you an F cell. So F cell, F positive cell means a bacteria that has its own genome and a plasmid. So the F plasmid. The HFR cell is basically when it has now the F plasmid integrated. So that's called HFR cell. And of course, this is just an example of the F plus, but we see this a lot with plasmids that encode for antibiotic resistance. Certain E. coli, for example, will have, or let's put Staphylococcus aureus. Staphylococcus aureus is a very nasty disease that can create very, how can I put it, septicemia, basically infection of the blood. It can be very lethal. So Staphylococcus aureus sometimes is very resistant to antibiotics. Some are and some are not. What happens is that in some of the Staphylococcus aureus that are resistant have these plasmids that encode for antibiotic resistance genes, genes that are able to stop antibiotics. And what can happen is that the Staphylococcus aureus can pass these resistance genes to other, how can I put it, Staphylococcus aureus cells that do not have this basically resistance, and then now they can make them resistant. Or they can pass also plasmids that are highly pathogenic. An example here is this E. coli. E. coli sometimes can take plasmids that actually helps them attach to your kidneys much better. So it's making them much more pathogenic. Pathogenic means much more causing disease. So they're giving them more disease traits. So they can get these plasmids that helps them attach to your kidneys better or to move to the kidneys even better. So certain genes or certain plasmids that can be transferred via basically conjugation can actually be a huge issue. Now, my final thing that I want to talk about is CRISPR. CRISPR is a very interesting thing. So one thing what happened is that when basically scientists started to sequence genomes of prokaryotes, they saw that there were regions in the DNA that had a lot of repeats. So they had a lot of repeats of certain basically sequences that were basically then of course kind of separated by spaces. So this hence was called CRISPR, clustered regularly interspaced short palindromic repeats. So there were certain repeats of like, let's say certain amino acid, sorry, DNA repeats that were basically spaced or like kind of separated by certain spacers, so random DNA. But then they found out that these repeats actually were DNA from viruses. So then they found out it's like, oh wow, this is in some sort of way an adaptive immune system. So adaptive immune system is basically a specific immune system of prokaryotes. So certain prokaryotes actually have these repeats of viruses in a way that basically they will recognize these viruses. So what happens is that basically these repeats will create RNA that degrades viral nucleic acid. So imagine this. You have a bacteria that has, for example, a CRISPR, so a palindromic repeat of the virus of, let's imagine, influenza. Okay? So if when influenza basically comes and infects this bacteria, the bacteria will not allow it because now it has its RNA, it has its DNA, so it's going to create RNA that's going to degrade it. So hence, it's very specific. So basically, this is very useful for gene editing in the labs and is being proposed as a tool for gene therapy. Because what we can do is we can use this CRISPR to actually help us basically remove any type of DNA. So in more the therapy session. So I'll be describing a bit this more in class, or I'll actually put a video that's gonna make this more understandable for you guys what CRISPR is, just for your actually let's put it this way. It's gonna help you understand what CRISPR is. We don't want you to fully memorize that, but just to know what basically CRISPR does. CRISPR provides the immune system an adaptive immune system towards prokaryotes against viruses.